Hello, everyone. Welcome to Human Biology. This is chapter 3. In this chapter, we are going to focus on cell structure and function. The child that you see on the picture here was born with a condition that we call the Tay-Sachs disease. The Tay-Sachs disease is a condition in which an enzyme is missing. That enzyme is specifically found in an organelle of the cell called lysosome. By that enzyme lacking, you have an accumulation of lipids in brain cells. This causes the cells, this causes the brain to degenerate and to cause the infant to not have coordinated movement. That is a major issue, only caused by an enzyme in the lysosome that is defective. So this is to show you the importance of knowing the cell, the structures, and the functions of these different structures. With that being said, let's dive in the chapter. As we dive in the chapter, I want you to think about a number of questions. The first one is, what is a cell? The next one is, why are most cells small? The third question is going to be, what are the different kind of cells that are found as we consider living organisms? What is common among those cells and what is different? How are cells organized? How do chemicals cross the plasma membrane, whether they're going in or out? What are enzymes and what is their role in metabolic reactions? And finally, what is cellular respiration? Now, in the first question, what is the cell? Note that a cell is defined as the basic unit of life, which means that the cell has all the characteristics of what makes life. They are able to reproduce, they are able to respond to environmental stimuli, they are able to adapt in a long-term manner. Also, all living organisms are made up of cells. That's another characteristic. And finally, a cell always comes from a pre-existing cell. These three components, that the cell is the basic unit of life. All living things are made up of cells, and a cell comes from a pre-existing cell, fall in what we call the cell theory. I expect you to know that. I will test you on that. Now, cells are in general small. Why are cells small? Cells are small because they need to create a chance for chemicals to come together for metabolic reactions to occur. If a cell is big, chance that the chemical are going to come together is low or slow, right? And what we notice is that the largest organisms take forever to reproduce, while the small one reproduce really quick. Say, for instance, bacteria. In 20 minutes, they can reproduce, right? They have a reproduction cycle that is about 20 minutes for some of them, right? While humans are going to take 20 year plus, maybe less, but take longer to reproduce. Our cells, if you consider, for instance, among our cells, our nerve cells are take longer to reproduce compared to our skin cells that renew all the time. Anyway, cells are small because of efficiency of reproduction, growth, metabolic activity. With that being said, for us to study cells, we need microscopes. In this course, there are two types of microscope we are going to focus on. The first one is the compound light microscope. What are some advantages of the compound light microscope, and what is a compound light microscope? A compound light microscope uses a light beam to view the object. Now, as they use a light beam to view the object, we are going to be limited as to the amount of details we can see using a light microscope. Now, the magnification that we can achieve, the highest magnification we can achieve with a good resolution, it's about 1,000 times. Magnification being an apparent increase in the object we are observing, right? Now, that 1,000 time magnification maximum, it's a limit as to how much detail we can see. However, the tremendous advantage that exists with a compound light microscope, it's that we can view specimens that are dead and specimens that are alive in their context, right? We can, for instance, take a drop of water from a pond and be able to see some protist swimming in that drop of water. Now, in addition to the compound light microscope, we are going to have the electron microscopes. Electron microscopes are characteristic by the fact that they are using an electron beam to be able to observe structures. This electron beam is accelerated with a very high current. Because it is accelerated with a very high current, it is not possible to observe live specimens, which is a downside. But the advantage, though, of an electron microscope is that we can magnify object in a very in such way that we can see very, very teeny tiny details. The maximum magnification we can reach, it's about 100,000 times. That's a lot, but that's very good because we can see object with very clear resolution, right? Now, there are two types of electron microscopes. We have a transmission electron microscope that allows us to see fine details inside of the cell. It allows us to have a 2D image of the object we are looking at. While the scanning electron microscopes works like a scanner, it allows us to see a 3D object. So it scans the surface and allows us to see the fine details of the surface. Everything else is very similar to the transmission electron microscope in terms of using electron beam to observe the object and being able to increase the magnification to up to 100,000 times. Now we have 3 micrographs here. A micrograph is a picture that we take with a microscope. We have on our right hand side a picture of a light with a light microscope that shows a blood vessel with red blood cell. You can see that the resolution in that picture with the light microscope is not as good as that of the middle that shows a micrograph taken with a transmission electron microscope, right? Where we see a blood vessel here with red blood cells. We have much more details compared to the light compound microscope. And also the third photograph, the scanning taken with the scanning electron microscope, we have red blood cells. Actually, we can see fine details on the surface of red blood cells. Now, when we look at the different types of cells across living systems, we see that there are two major types of cells. We have prokaryotic cells and eukaryotic cells. Prokaryotic cells are very simple cells in the sense that they don't have very complex organelles inside of them. They are thought to be the first cells to have existed, and from these cells, all the cells have evolved, including eukaryotic cells. Now, prokaryotic cells typically do not have a nucleus. Their genetic information is enclosed in an area of the cytoplasm that we call the nucleoid. Right? So if I was to draw a bacterium, this would be the limit of the cell. This would be the DNA, right? So, and this would be the cytoplasm here. Okay? So here we have cytoplasm. Cytoplasm. Here we have DNA. And here we have plasma membrane. All right. Then eukaryotic cells. Well, actually, before we go to eukaryotic cells, prokaryotic cells are going to include bacteria and archaea. These are simple cells. Eukaryotic cells are going to be characterized by the fact that they are much larger most of the time. And in addition, they're going to have a true nucleus. That nucleus will contain the DNA, which carries the genetic information. In addition to that, we're going to have many membranes. Membranes that are going to limit the physical space of organelles. Organelles being the structures with very specific activities in the cell. One particular organelle, such as mitochondrion, produces ATP in large amount. There is no other organelle in animal cell that produce ATP, like mitochondria, right? So as we look at prokaryotic cells versus eukaryotic cells, what is similar among them? Few things are similar. Actually, I should say 4 things are similar. First and foremost, they all have a structure that sets the limit between the inside and the outside of the cell. That structure is called the plasma membrane. Then we are going to have the cytoplasm. The cytoplasm is where everything that is inside of the cell is going to bath. It's a semi-fluid substance, all right, in which organelles are going to be found. And then we are going to have DNA. DNA is going to be the molecule that houses the genetic information that causes the cell to exist. And finally, but not least, I would like to mention ribosomes. Ribosomes. Ribosomes are responsible for protein synthesis. Protein synthesis. Now, here we have a representation of a cell, an animal cell. That cell has all the structures you could find in a cell, in an animal cell, like a skin cell, right? We have the plasma membrane. We're going to have the nucleus. We're going to have some endomembrane system, including the endoplasmic reticulum, the Golgi apparatus, mitochondria. We're going to have centrioles, centrosomes, transport vesicle. We're going to have the cytoskeleton. Now, just looking at this slide, you can actually learn everything you need to learn about cell structure and function. But let's move on. Now, the first step we are going to make is going to be the plasma membrane. What is the structure of the plasma membrane? The plasma membrane is going to be made up of a number of molecules, the most abundant being a phospholipid bilayer. That phospholipid bilayer is going to develop obviously from phospholipids and is going to cause a limit as to what can come in and what can go out. So much that the phospholipid bilayer is said to be selectively permeable, or the plasma membrane is said to be selectively permeable. What else is found in the plasma membrane? Proteins. Okay? These proteins are embedded in the plasma membrane. These proteins play multiple roles. We're going to discuss that later. And finally, we are going to have cholesterol that play the role of support, and carbohydrates are going to be attached to proteins and lipids of the plasma membrane. So here we do have a graphic representation of the plasma membrane with the phospholipid bilayer. Please review that. The proteins embedded in the plasma membrane, these are integral proteins. We have cholesterol. We have some proteins that are not directly attached to the plasma membrane, but that are adsorbed to the plasma membrane. We say that these proteins are peripheral proteins. We have some carbohydrates that are attached to proteins, like this carbohydrate here. When a carbohydrate is attached to a protein, the protein along with the carbohydrate is now called glycoprotein. We have the term glycoprotein here. And when carbohydrates are attached to lipids, like in this instance here, we have a glycolipid. Glyco means sugar, right? Glycolipid, a lipid to which a sugar is attached. Glycoprotein, a protein to which a sugar is attached. Okay? What else should I say here? Outside of the cell, you are going to have many chemical components. These many chemical components fall in the generic term of extracellular matrix. That extracellular matrix is going to be used as a substrate for attachment of cells so that cells can come together to form tissues. Inside of the cell, close to the plasma membrane, we're going to have cytoskeletal proteins or cytoskeletal filaments. Now, I said earlier that the plasma membrane is semi-permeable, what means that some chemicals can cross freely the plasma membrane while other chemicals have a hard time crossing the plasma membrane. But what can cross freely and what cannot cross freely? Well, what can cross freely are chemicals that are small, non-charged, non-polar. Okay? So some examples are oxygen gas, carbon dioxide gas, and nitrogen gas. These three gases are able to cross freely the plasma membrane. Now, because most of the chemicals in our system are either charged or large, these chemicals are not going to typically cross the plasma membrane. Then you understand the concept of selective permeability of the plasma membrane, right? Now, the proteins that are embedded in the plasma membrane are going to facilitate the movement of some chemicals in and out. For instance, sugar. How do we import sugar in? Using some proteins that are embedded in the plasma membrane. Believe it or not, even water to get in or out of the cell needs to use some proteins that are embedded in the plasma membrane. One such example is aquaporin. Aquaporin is the protein in charge of allowing water in and out. So, with that being said, what are the different mechanisms, the different ways by which chemicals cross the plasma membrane? One is diffusion. The other one is osmosis. The third one is facilitated transport. Fourth one, active transport. And finally, exocytosis and endocytosis. Now, let's first of all look at diffusion. What is diffusion? When it comes to human biology and biology in general, what we call diffusion, it's basically the movement of chemicals but water across the plasma membrane. And that random movement of molecule is going to be from compartment of high concentration toward compartment of lower concentration. The movement of molecule, the net movement is going to stop when we have an equilibrium on both sides. Now, we know that our body is an environment that is water-based. Therefore, water is the solvent of life. Anything else is a solute. Whether you are talking about salts, or you are talking about carbohydrates, or proteins, or nucleic acids, all of that is dissolved in a water-based environment. And anything that is small, non-charged, at that point is going to be able to do diffusion, right? Such as oxygen gas when we breathe it in, and carbon dioxide when we breathe it out. Anyway, because most chemicals cannot cross the plasma membrane, water is the one that will have to cross the plasma membrane. How is water crossing the plasma membrane? By a mechanism that we call osmosis. Osmosis is simply the diffusion of water across the plasma membrane. All right? So I want you to remember that. Now, diffusion is going to be a way for molecules that are high concentrated in one compartment to move in a compartment where they are less concentrated, so that at the end of the day, we have a balance. So when we consider this area here, we can clearly see that water is more concentrated here and the solutes are more concentrated here than they are here. Therefore, over time, both chemicals are going to move across the plasma membrane until an equilibrium is reached. And this is when diffusion is going to stop. But in reality, diffusion never really stops. You only have as many molecules that goes on the left side as molecules that go on the right side. Now, because water is the one that moves across our cells in the context of our body, we will have to ask the following question. In which context is our cell at a given stage of our life, at a given stage of the day? Ideally, the body wants to keep the cell in what we call an isotonic environment. In an isotonic environment, if you consider a solute such as sodium chloride, NaCl, that sodium chloride is dissolved in water both inside of the cell and outside of the cell. Now, in an isotonic environment, we would have concentration of sodium chloride the same in the solute, in the solution, and inside of the cell. In that situation, the cell is going to be in a physiologically acceptable environment for metabolic processes to take place. However, this is not always, that ideal situation is not always happening in our systems, so that the cell can be in an environment where the solute NaCl is more concentrated inside of the cell than it is outside of the cell, in which case we say that the cells are in a hypotonic environment. Hypo means less. In a hypotonic environment, there is a need for water inside of the cell. When water rushes inside of the cells are going to take up volume. Now, because animal cells don't have a cell wall, if nothing is done, these cells can actually burst, right? Which means that physiologically, it's not a good thing that our cells be in a hypotonic environment. Now, on the other hand, and on the opposite side of the spectrum, the cell can find itself in an environment where the solute NaCl is so concentrated outside that water is going to leave the cell to go outside to try to keep things at balance. This is causing the cells to shrivel, right? By shrinking like that, the chemicals or the metabolites are so close to each other that the metabolism is not going to happen properly, which is going to cause the cell to die or stop all metabolic processes. That's typically what happens when we go hiking. We feel the need to drink water. We feel the need to drink water because by sweating, we are losing water. And our body, the cells in our body feel that they are in a hypertonic environment. Therefore, we need to drink water to compensate, right? All right. So we talked about diffusion and osmosis. How about facilitated transport? Now, most molecules, again, are either charged or large. Because of that, molecules are going to get in the cell through the plasma membrane only by using proteins that are embedded in the plasma membrane. These proteins are going to be selective of the kind of molecules they can allow through. Proteins are selective of the kind of molecules they can allow through. Now, facilitated transport, it's a type of transport system that does not require use of energy, but it does require the presence of a transporter or a carrier. All right? It requires the presence of a transporter or a carrier. In a facilitated transport, the molecules are moving from compartment of high concentration toward a compartment of low concentration. That's why you have the green molecule here going from the outside toward the inside. The net movement is going to stop when you have equal concentration on both sides, inside and outside. Now, remember that facilitated transport does not use energy. Very important to remember. And that facilitated transport is also called facilitated diffusion. Facilitated diffusion, right? It's also called passive transport. Why passive? Because it does not use energy. as opposition to passive transport, you are going to have active transport. Active transport is a type of transport that requires use of energy. That energy is in the form of ATP. ATP is going to activate a transporter. By activating a transporter, this is going to cause a transporter to allow chemicals in or out. Now, what is characteristic of active transport? It's that because of the expense of energy, the transporter can actually carry molecules against their concentration. In other words, even though a chemical is already abundant in one particular compartment, the cell can still bring that chemical in that compartment, right? So here we have an example of a few chemicals that are brought in by active transport. We have potassium ion and sodium ion. While potassium ion is transported inside of the cell, sodium ion is transported outside of the cell. This process requires expense of an ATP molecule that is broken down into ADP and a phosphate molecule that is transferred to the transporter. All right, other type of ways molecules or elements can cross the plasma membrane. We have endocytosis. Endocytosis is a form of transport that allows the cell to bring some particles in. These particles can be solid particles such as bacteria. Imagine that you are fighting an infection, and that infection is strep throat, right? So this solid particle will be a streptococcus, right? That streptococcus can be engulfed in, brought in by a process that we call phagocytosis, which is actually, by the way, cellular eating. That cellular eating is actually inspired of what we see happening with some protists, such as, what are they called? Amoeba. Okay? So when amoeba drink, we call this pinocytosis. So there is a cellular process by which we can bring some droplets inside of the cell by way of pinocytosis or cellular drinking. There is another endocytosis that requires the presence of receptors. When chemicals need to be brought in, this chemical will have to attach on a receptor. When a certain number of receptors have chemicals attached to them, there is a threshold by which some invagination is triggered so that you have a vesicle that is formed. That vesicle is formed because of these receptors and the cargo to bring in. This is called receptor-mediated endocytosis. These three forms of endocytosis, meaning phagocytosis, pinocytosis, and receptor-mediated endocytosis, these three forms require expense of energy or ATP. No ATP, no phagocytosis, no pinocytosis, no receptor-mediated endocytosis. I want you to remember that. Finally, but not least, exocytosis. Exocytosis is the inverse process compared to endocytosis. Exocytosis is going to take material from in toward outside. Imagine, for instance, when a cell synthesizes antibodies. These antibodies that are synthesized inside of the cell have to be released outside of the cell. These antibodies are going to be released by exocytosis. Now let's take a look at cell compartments. Among the cell compartments, we are going to have the nucleus. After the nucleus, all the organelles we're going to cover, ribosomes, and the endomembrane system. Let's first of all stop on nucleus. The nucleus is going to be made up of a number of parts. First, the nuclear envelope. The nuclear envelope is going to have pores. These pores are holes through which material is going to go out of the nucleus and come inside the nucleus. And inside the nucleus, we're going to have the nucleoplasm. The nucleoplasm is going to contain DNA, proteins, and carbohydrates, a bunch of things. Now, DNA is not just in the form of DNA, it's combined with protein and form a structure that we call chromatin. Chromatin can condense and form chromosomes. Now, beyond that, you do have the nucleolus. The nucleolus, it's a part of the nucleus that is dense, where ribosomal RNA are going to be produced. This is also where ribosomes start their assembly. The assembly is not completed there, but it starts there. Now, the ribosomes are going to be those organelles that are made up of ribosomal RNA and proteins. These ribosomes actually in our human cells, they occur in large subunit ribosome and small subunit ribosome. The large subunit ribosome is called not 80S. It's called 60S ribosomes, while the small subunit of the ribosome is called 40S subunit. This is the large subunit. This is the small subunit. Together, large subunit and small subunit are going to be called 80S ribosome. But let me close that parenthesis. So, the ribosomes are specialized in protein synthesis. They are the site of protein synthesis. The ribosomes are going to be found free-floating in the cytosol, or they're going to be attached on the membrane of the endoplasmic reticulum, causing the endoplasmic reticulum to look rough. That's why we would call rough endoplasmic reticulum. Okay, and that rough endoplasmic reticulum will be involved in protein synthesis. Now, here we have the nuclear envelope, as you can see here. It's a double membrane, but I spare you the detail. We're going to have here the pores, the nuclear pores. All right, we're going to have chromatin in here. We're going to have ribosomes in yellow that are attached to the envelope, but they are also attached here on the surface of the endoplasmic reticulum, causing the endoplasmic reticulum to look rough. That rough endoplasmic reticulum, because it has proteins attached to them, they are going to be involved in protein synthesis, right? The smooth endoplasmic reticulum does not have ribosomes attached to them. Therefore, they are not involved in protein synthesis. Notice that the nuclear envelope expands to the rough endoplasmic reticulum, which itself expands to the smooth endoplasmic reticulum, which shows you that this is an endomembrane, a network of membrane in the cell. I want you to remember that. So here we have a micrograph of nuclear pores, and here a micrograph of the rough endoplasmic reticulum and smooth endoplasmic reticulum here. Now, what is the endomembrane system? The endomembrane system is this network of flattened sacs. This network of flattened sacs is going to allow organelles to have a specialization in the type of chemical reactions they are catabolizing, they are getting involved in, right? So this membrane system is going to include the nuclear envelope and the endoplasmic reticulum. One I have not mentioned so far is the Golgi apparatus, and others I've not mentioned so far are the lysosomes and the transport vesicles or other vesicles, right? I already showed you how the nuclear envelope extends to or expands to become the endoplasmic reticulum, okay? But know that the endoplasmic reticulum itself is going to expand to become the Golgi apparatus, okay? And from the Golgi apparatus, you have lysosomes that are going to form, vesicles that forms, and vesicles that forms from the endoplasmic reticulum and from the Golgi apparatus. Anyway, so let's take a look at here how the endomembrane system functions. Say you have a gene in here. That gene is expressed. We have a messenger RNA. That messenger RNA that is produced from DNA is now going to be converted into protein. The ribosomes that are attached here are the ones that are going to be responsible for converting this messenger RNA into proteins. These proteins are going to be released in the inner space of the rough endoplasmic reticulum. The purpose of that is for the protein to be modified in the rough endoplasmic reticulum and to be folded. After the modification, a transport vesicle is going to pinch off, going to migrate to the Golgi apparatus. In the Golgi apparatus, the protein is going to be further modified. Typically, they're going to be modified with addition of sugar residues. Beyond that modification of the protein with an addition of a sugar group, the protein is going to be folded to its final shape and packaged so that it's going to be delivered to the final destination. There are three possible outcomes to these proteins that are synthesized through the rough endoplasmic reticulum. One is that these proteins can end up outside of the cell, right? A good example of that, it's an antibody that is produced by a white blood cell that is released outside of the cell. That is a good example. Another fate, it's that the protein can be part of integral proteins. You remember when we looked at the plasma membrane, we said that some plasma membrane are embedded, have some proteins embedded in the plasma membrane, some playing the role of receptors, right? Others playing the role of channels, other proteins playing the role of enzymes, right? How does this protein end up in there? Well, the protein end up in there because they are synthesized through the rough endoplasmic reticulum, then they go in the Golgi apparatus, and then they become part of the membrane of the vesicle that is going to merge with the plasma membrane and finally give something like this. That's fate number 2. Fate number 3 is that the protein can end up being an enzyme. That enzyme is going to end up in a little sac that we call lysosome. The lysosome is going to contain bunch of digestive enzymes. This digestive enzyme break about anything. Carbohydrates, complex and simple, it doesn't matter. Lipids, nucleic acids, proteins, you name it. They break down anything. So this sac-containing digestive enzyme is going to be very handy when it comes to fighting infections, because assume that you have a strep throat and that your white blood cell engulf the strep throat by a process that you see right here, phagocytosis. So the phagosome here, let me spell that, phagosome. Phagosome. The phagosome here is now going to migrate and merge with the lysosome so that we have what we call a phagolysosome. Phagolysosome. That phagolysosome is now going to cause a digestion, a breakdown of the bacteria that came in, and now you are safe, right? So 3 fates to the protein that are synthesized through the endomembrane system. 1 is to send the protein outside of the cell. 2 is to cause the protein to be part of the plasma membrane. 3 is to cause the protein that is actually an enzyme to be part of the lysosome. Please remember that. So that was for the rough endoplasmic reticulum. How about the smooth endoplasmic reticulum? The smooth endoplasmic reticulum is very much involved in the synthesis of lipids. It's also involved in the synthesis of carbohydrates and in detoxification. Everything really depends on the type of cell, where in the body is that cell located. In the liver cell, for instance, the smooth endoplasmic reticulum does a lot of detoxification. All the drugs that we take when we are sick are discarded by the smooth endoplasmic reticulum. In muscle cells, the smooth endoplasmic reticulum is the area where calcium ions, Ca2+, are stored. And calcium ions are essential for contraction of muscles. Let me close that parenthesis. So the rough endoplasmic reticulum, I already told you that it had ribosomes attached to them. Because of those ribosomes, the rough endoplasmic reticulum is going to be involved in processing proteins. The smooth endoplasmic reticulum, that one is going to lack ribosomes, is going to be involved in making carbohydrates, lipids, and depending on the cell, is going to be involved in storage of calcium, for instance, or detoxification. How about the Golgi apparatus? The Golgi apparatus is going to be essential for processing protein by adding sugars to them, packaging these proteins, and delivering these proteins where they need to go. Now, think a minute. This Golgi apparatus works exactly like a post office. These are the guys that are in charge of distributing the mail where they need to go. Now, if there is a mislabeling in the Golgi apparatus, the fate of the protein may not be what it should have been. I was telling you about the Tay-Sachs disease at the very beginning of the chapter. Well, the enzyme that is lacking in the lysosome is not in the lysosome because in the case of the Tay-Sachs disease, the Golgi apparatus is not doing the job it is supposed to do for that enzyme to end up in as part of the lysosome. Let me close that parenthesis. All right, the lysosomes are going to contain digestive enzymes, and lysosomes are formed from the Golgi apparatus. And the vesicles are going to be of different kind. They are going to be play the role of transport vehicles in the cell. And now, the cytoskeleton. The cytoskeleton is going to be essential to provide a shape to the cell, to allow the cell to organize the organelles in the cell, what we call cytoplasmic streaming, and to allow movement. There are three different types of cytoskeletal fibers. We have the microtubules, we have the actin filaments or microfilaments, and we have the intermediate filaments. They all have different roles. Now, what is the role of microtubules? Microtubules play very important roles, including in our human cells, including in cell division. When our cells divide, they will have to distribute the genetic information among the two nodal cells. Microtubules are the one that take care of this, right? Other role of microtubules, when we consider the sperm cells have an appendix that is called flagellum. That flagellum actually is made up of microtubules. These microtubules are going to be activated by ATP and create a whip-like motion which allows the flagellum to swim. The sperm to swim. Now, that's for the flagellum. How about the cilia? The cilia are found in part of our body, including along the windpipe, where they are going to vibrate and cause the mucus to move from one direction to another. Now, these cilia are going to have microtubules, and with ATP, these cilia are going to cause motion that allow, wave-like motion that allow the mucus to move on the surface of the epithelium. Now, we talked about the cell structures, both the structures inside and a little bit about the extracellular matrix. Now, the cells come together to form tissues. The question is, how are these cells coming together? Sometimes cells are going to come together by coming very closely attached together. Now, let me be a little more specific. The junctions that we found between cells occur in three kinds. Three major types of junction. One is the kind of junction that we find between skin cells. That kind of junctions are kind of a little loose. They are called adhesion junctions. They also carry another name. It's desmosomes. Desmosomes. The desmosomes are adhesion junctions, right? These are going to be very mechanical junctions, right? Like in the skin. Another type of junction, it's the tight junctions. The tight junctions are going to be found between cells to make sure that where there is fluid, the fluid doesn't leak through. The junctions are going to develop because of a zip-like structure, as you can see here. Zip-like structure. This zip-like structure allows two neighboring cells to be closely tied together. This is, for instance, what we are going to find in the digestive tract. You know that the stomach, for instance, is holding some fluid, right? The small intestine is holding some fluid. Now, how about the bladder, right? The urinary bladder holding some fluid. You need some tight junctions there. And then the last type of junction that we are going to address is going to be the gap junctions. The gap junctions are going to occur in tissues where there is a need for communication between neighboring cells. If you consider, for instance, the heart beats as a syncytium, right? When a signal, an impulse comes, the cells that first receive the impulse does not contract and then the other one contract next. No. The information comes in the first cell. Before that cell can develop a response, that information has already traveled throughout the other muscle cells, so that the heart beats as a unit. Okay? And this is possible because of gap junctions. Now, we saw how cells come together to form tissues, the different types of junctions. At this point, we are going to dive in metabolism. When we talk about metabolism, I want you to think about two things. One, reactions of synthesis. Two, reactions of hydrolysis. Reactions of synthesis in our body are also called anabolic reactions or anabolism. Reactions of breaking down or reactions of hydrolysis are also called catabolic reactions or catabolism. What is metabolism? Metabolism is the sum of all anabolic reaction plus all catabolic reactions that are happening in your system. In other words, reactions of synthesis and breaking down. These reactions of synthesis and breaking down hardly happen in one single step. These reactions happen in a multiple step type of way of doing things. It's a process. That process is going to involve steps. Step 1 or reaction 1, reaction 2, reaction 3, reaction 4, reaction 5, 6, and so on and so forth. So that when you want to synthesize, for instance, an amino acid, the system is going to start with a substrate, right? A reactant. That reactant is going to undergo a series of chemical reaction until you have, at the end of the line, production of an amino acid. It's a process. So here we have series of chemical reactions that are happening here. Each series of chemical reaction is going to involve a substrate and, at the end, a product. The substrate typically is a reactant. The product is the result of the chemical reaction, right? Now, this series of chemical reactions, we are going to study some of them in this chapter. Now, before we dive into some of them, know that all these reactions that occur in anabolism and catabolism, even though these reactions are intended to occur, they do not just occur by themselves. They occur with the help. The help of what? The help of enzymes. What are enzymes? Enzymes are catalysts. They are proteins. These enzymes that are proteins are going to target very specific substrates. An enzyme that catalyzes the reaction of breaking down glucose will not know how to catalyze the reactions of breaking down a fatty acid. Enzymes are proteins. Their 3D shape matters. Their environmental conditions in which they are evolving matters, which means that a drastic change in the pH can affect the activity of enzyme. A drastic change in the temperature can affect the activity of an enzyme. Enzymes are going to recognize very specific substrate. This substrate are going to bind on specific areas of the enzyme called active site. When an enzyme interacts with a substrate through the active site, this is when catalysis of the reaction is going to be made possible. When we talk about catalysis of the chemical reaction, really what we mean here is that the enzyme is going to help a chemical reaction occur. But question is, how does that enzyme helps? Now, an enzyme doesn't change an outcome of a reaction. Let this be clear. An enzyme does not change the outcome of a reaction. An enzyme does not make a reaction that is impossible possible. No, an enzyme cannot do that. Enzyme can be used over and over. Other thing is that every single chemical reaction that occurs in our system is intended to occur. The question is at which pace? Question also is what limits the occurrence of these chemical reactions? What limits these chemical reactions? It's actually the requirement of energy. Does the system have the amount of energy for that chemical reaction to occur? It's a yes or no question. Okay? So, if you answer no to that question, the chemical reaction is not happening. If you answer yes, then the chemical reaction is happening. The amount of energy that is required for a chemical reaction to occur is called activation energy. That activation energy works like a price tag, right? A given reaction has a certain amount, requires a certain amount of energy for that chemical reaction to happen in our system. What enzymes are going to do is to actually reduce that required amount of energy. So enzymes are going to speed up chemical reactions by lowering the necessary activation energy for that reaction to occur. I want you to remember that, and I will test you on that. Now, most enzymes are controlled in our system. How are these enzymes controlled? By controlling the availability of some non-protein molecules that are called coenzymes. Some examples of coenzymes are a molecule that is called NAD+. Another one, it's NADP+. Another one is FAD. Now, these are all coenzymes. But in our system, not only coenzymes control the activity of enzymes. Other things that control activity of enzymes are cofactors. Cofactors. By the way, coenzymes are cofactors, but the coenzymes are cofactors that are organic cofactors. Organic cofactors. Organic cofactors. Why do I say organic cofactor? Because there are some cofactors that are not organic. If you think, for instance, of zinc, you think of calcium, you think of iron. These are all chemicals that can react with enzyme to aid enzyme in becoming fully active. Okay? Let me close that parenthesis. So we have a substrate here. That substrate, see how the substrate is very similar to the active site. So this enzyme is going to be able to catalyze the chemical reactions of breaking down this chemical here. Now you have the formation of a complex enzyme substrate here, so that the chemical is going to be broken down here, so that we have a product that consists of two chemicals here. And at the end of the day, the enzyme is restored and ready to catalyze another reaction. That's for breaking down. For synthesis, an enzyme is going to bring two substrates together, okay? But this substrate have to match the active site. If there is no match, no catalyzing any chemical reaction, then you have a complex that forms right here. That complex enzyme substrate is going to cause the production of a product here, and the enzyme is released at the end of the day, and we have an enzyme that is ready to do its work one more time. Now, I was telling you that a given chemical reaction requires a certain amount of energy that we call activation energy. Without that amount of energy, no chemical reaction is happening. How do enzymes speed up chemical reactions? By lowering that activation energy, which is going to cause the cell to save energy and to be able to maintain metabolic activity without really having to go in a dormancy kind of mode. Now, what is an example of metabolic process? An example of metabolic process is going to be cellular respiration. Cellular respiration occurs in mitochondria. The mitochondria is the powerhouse of the cell. The mitochondria, it's an organelle that has a double membrane, an outer membrane, and an inner membrane. That inner membrane is folded with structures that are called cristae. And that mitochondrion is going to be one of the unique is going to have unique features, that they're going to have some DNA. They do have some DNA. And they do have some ribosomes. But let me close that parenthesis. Let me focus on the fact that the mitochondrion is the powerhouse of the cell. That's the guy that ultimately produces ATP. If something happens to the mitochondrion, such as when you ingest a poison, such as cyanide, well, you are in bad shape, because cyanide shuts down mitochondria by interfering with ATP production. You don't have enough ATP, and this is how you die easily when you ingest cyanide. So that's why cyanide is a bad news. So, mitochondria resemble bacteria in the sense that their structural organization is very similar to that of bacteria. 1, 2, they have DNA, just like bacteria. 3, their ribosomes are very similar to bacterial ribosomes. I'm not going to bug you with the details. This is human biology and an introductory course. I'm not going to go in the details. Let me close that parenthesis. But let me just tell you that because they resemble so much bacteria or prokaryotes, there is an idea that eukaryotic cells or a larger cell had engulfed this prokaryotic cell so that over time they became interdependent. This is the theory of endosymbiosis. I'm not going to bug you with that. Just for your information, I'm just writing here endosymbiosis. Endosymbiosis. Look it up. But I'm not going to test you on that. Now, cellular respiration, what is it? Cellular respiration, it's a cellular process by which ATP is produced in the cell. Now, that cellular respiration occurs in many steps that include series of chemical reaction. 3 major steps. 1 is glycolysis. 2 is the citric acid cycle. 3 is the electron transport chain. In every single stage, you have production of ATP. The only thing is going to be how much ATP, right? That's something we are going to take a look at. Now, say you had a breakfast. You digest your breakfast. Ultimately, you have some glucose in the blood. That glucose is going to leave the blood, and through some transporters here, that glucose gets inside of the cell. That glucose is going to undergo glycolysis. Glycolysis, it's literally means the lysis of glucose or the lysis of a sugar. So breaking down a sugar. When that sugar is broken down, glucose is converted into a chemical that is called pyruvate. In the process, you have 2 net ATPs that are produced. Okay, forget this slide. This arrow here. So you have 2 ATPs that are produced. 2 net ATPs, not 2 ATPs, but 2 net ATPs are produced here. But along with these 2 net ATPs, you also have something else that is produced. It's a molecule called NADH. That molecule called NADH, it's an electron acceptor. It catches electrons that come from glucose and get them to NAD to become NADH. That's one thing. And then, beyond glycolysis, we are going to have the citric acid cycle and the electron transport chain. Notice that glycolysis occurs in the cytoplasm, while the citric acid cycle and electron transport chain occur in the mitochondrion. Now, let's go in the mitochondrion. In the mitochondrion, the byproduct of glycolysis, so pyruvate, is going to be broken down and go in the citric acid cycle. In that cycle, we have many NADH that are going to be produced, but beside NADH, there is another guy that I want you to remember. It's FADH2. These 2 molecules are cousins, but don't worry about that. I want you to remember that NADH and FADH2 are produced in the citric acid cycle. And remember that these are electron acceptors. They carry electrons that are very unstable, right? In the process, though, of the citric acid cycle, you have release of carbon dioxide and production of 2 ATPs. Now, the electron transport chain here is going to depend tightly on the presence of oxygen. Without oxygen, the electron transport chain is not working. The electron transport chain is going to pass electrons from NADH to the electron transporters, FADH2 to the electron transporters. In the process, oxygen is going to accept those electrons, and oxygen is going to produce water, H2O, and ATP is going to be produced in the process. I spare you the details. Notice that you have 32 ATPs that are produced. Okay? that tells you how much ATP is produced in this process of cellular respiration in the mitochondria. Now, in the process of glycolysis, we have that this happens in the cytoplasm, breaks down glucose into 2-pyruvate molecules, produces NADH, which is an electron carrier, right? An electron acceptors, and 2 net ATPs that are produced. This process does not require oxygen. The citric acid cycle, it's a cycle that occurs in mitochondria. It produces NADH, but also FADH2. I want you to remember that. And 2 ATPs are produced. In the process, you have release of carbon dioxide, CO2. Now, beyond that, electrons are now the one that are going to come into play. So here, electrons from NADH and FADH2 are going to be passed down from one electron carrier to another electron carrier. In the process, these electrons are going to be deprived of their energy. That energy is going to go toward the production of ATP. At the end of the day, production of 32 to 34 ATPs are going to be made in this process. This process requires energy. This process requires oxygen as the final electron acceptor. Now, here is the thing, and something I want you to remember. Imagine that you have a business. In that business, you are producing $2 million, 2 ATPs represent $2 million. Another $2 million here, ATP, and another $32 million ATP. So you have altogether 36 ATP, $36 million. Now, imagine that because of COVID, your business is not producing anymore this and this, which actually cause you to lose $34 million. Question: what is the likelihood that your business is going to survive if you go from $36 million to $2 million? Answer: chance that your business is going to survive is close to 0, if not 0, right? The reason why I'm showing you this, it's because when we ingest a poison such as cyanide, it's basically the equivalent of shutting down our mitochondria. These ATPs are not produced anymore, and we want to rely exclusively on glycolysis. This is not sustainable. That's why we die within 6 minutes when we ingest a poison such as cyanide, right? So, anyway, I want you to remember that oxygen is required for all reactions that happen in mitochondria. However, oxygen is not required for reactions that happen in the cytoplasm. So, beside the metabolism of simple sugars such as glucose, you have other carbohydrates that can be broken down to produce energy. Proteins can be a source of energy, but these proteins have to be broken down into amino acids first. From amino acids, now we can obtain ATP out of these amino acids, there is a complex series of chemical reaction, including a process that we call deamination, right? I'm not going to bug you with the details. For the lipids, if you have a triglyceride, that triglyceride have to, using lipases, be digested to produce glycerol and 3-fatty acid molecule. Glycerol is going to enter cellular respiration. The fatty acids are going to enter cellular respiration through the process of beta oxidation first that occurs in some organelles that we call peroxisomes. But let me close that parenthesis. Once you have byproduct of that beta oxidation, now you are going to have cellular respiration that is going to allow you to produce ATP. The bottom line is that not only glucose allows you to produce ATP, or the sugars allow you to produce ATP, proteins allow you to produce ATP, breaking down lipids allow you to produce ATP. Finally, but not least, what if we don't have enough oxygen, such as when we exercise? When we run and when we do an exercise, such as when we are running, at the beginning, we are good, we can go fast, and so on. But at some point, we start feeling fatigue. What causes to feel fatigue? We feel fatigue because the amount of oxygen that is needed is not, our body is not receiving enough oxygen. At that point, overall, our muscles are going to enter a type of metabolism that is fermentation. So fermentation occurs in the cytoplasm. This process does not require oxygen, one, and relies extensively on glycolysis. Glycolysis that produces only 2 net ATPs for 1 molecule of glucose that is produced. Now, in the process of fermentation, you have production of a chemical called lactate. That lactate accumulates in our muscle and cause that feeling of burning. All right? And that feeling of burning is going to basically cause us at some point, with the lack of energy, to basically say, okay, I'm done, I'm stopping, right? So fermentation, it's not a process that can allow us to sustain our physical activity. So this is where our chapter is going to stop. Thank you for your attention.